Our next speaker, Ardem Patapoutian, also was born by the sea in Eastern Mediterranean, in Beirut, Lebanon, to an Armenian family. Ardem later immigrated to the US, and he studied first his bachelor's at UCLA, and then he did his PhD at Caltech in the lab of Barbara Wold, where he was actually looking at transcriptional regulation during development, specifically muscle differentiation. Later, during his postdoc, he joined UCSF in the lab of Louis Reichhardt, where he investigated developmental programs leading to specificity of somatosensor neurons that encode touch and pain. Ardem is currently a professor at the Department of Neuroscience at the Scripps Institute in San Diego, and he's an investigator of Howard Hughes Medical Institute. Please join me. Welcome, Ardem.
Hi, everybody. Thanks for this very nice, cozy room. You look great. So I was just going to give you an overview of our work that was recognized by the Kavli Prize. And our perception of reality is, of course, very much dependent on our senses. And over the last few decades, we have learned quite a bit about how we smell, taste, and see. But this somatosensory system that David nicely introduced about how we gather mechanical, thermal, and chemical information from the environment and synthesize this for our sensory neurons to understand has been less known and less understood. But beyond the sensory system, most of communication within our bodies for our cells is done through chemicals, and yet how we sense physical forces beyond the nervous system is also not well understood. So I'm here mainly to tell you that your sense of touch is amazing. You are able to have nanoscale level of discriminatory power for your sense of touch. Beyond this, we rely on affective touch for our emotional health. And finally, for the related sense of proprioception, can tell you where your arms and legs are in space without looking at them, for doing simple tasks like walking, but also more complex ones like playing musical instruments without looking at your fingers, beautifully demonstrated here by Emmanuel Axe and Yo-Yo Ma. But my absolute favorite image that shows the power of touch is this one. This is in Washington, D.C., in the White House, and this little boy has visited President Obama, and he tells him that his friends tell him his hair just looks exactly like Obama's. And Barack, of course, just immediately bends and says, "Why don't you touch it?" And so we're not just talking about the able to discriminate the texture of his hair, but the very powerful nature of affective touch. And Barack knows that although people say seeing is believing, touching is also very important for this. So the flip side of touch is pain, and acute pain is absolutely essential for our survival, but chronic pain, usually defined as pain that persists even after the stimulus is gone, is still a major unmet medical need. Just look at the opioid epidemic the last few years that was well advertised. So all of these mechanosensory phenomena that I talked about, touch, proprioception, pain, all initiate in this group of peripheral nervous system, sensory neurons, just dorsal spinal cord, that gather information from the periphery and send this information to the spinal cord and to the brain. And one of the characteristics for these neurons is that they're extremely heterogeneous. As color-coded here, some are low threshold and sense touch and proprioception. Others are focused on sensing temperature and pain. And others are these proprioceptors that we'll talk about. And if you follow these neurons to the periphery, they also distinguish themselves by what they innervate. For example, in red, proprioceptors innervate muscle spindles, and many other neurons innervate the skin. That there are a variety of different anatomical endings, including Merkel cell complexes, Meissner corpuscles. And the information, the mechanical information you get from all of this are synthesized in your brain, what you know as touch. In addition, again, David touched upon this idea of interoception, which is innervation of internal organs. The RGs also do this, and we'll talk about it as well. So how about the molecules? How do you sense a gentle breeze, or in this case, a cactus pricking your finger? Unlike other ion channels that we know that are chemical sensitive, it was known for decades that there are ion channels that are specifically sensitive to acutely respond to physical forces, such as temperature and mechanical force, but their identity remained unknown for quite a long time. So David Julius's lab, my lab, and others found trip-ion channels that are involved in thermosensation from 1997 on. But just 12 years ago, the identity of mechanically activated ion channels responsible for touch, pain, and all other forms of mechanosensation in vertebrates was still unknown. Just a little bit of history on these ion channels. So the first indication that they even exist came from Jim Hatzbad's lab, previous Kavli winner, and his work was in the hearing field, and he showed that the response time between mechanical stimulation and the first recording of hair cell transduction for hearing was so fast that it could not happen through second messenger system, such as GPCR signaling that's prevalent for vision and taste and smell, for example. So it had to be an ion channel directly gated by mechanical force. So these types of channels were shown to be active in many different cell types a few years later by Fred Sachs, that showed that he could record from stretch-activated channels from muscle cells. But what's the identity of these ion channels? Throughout the years, many candidates and receptors have been identified in bacteria, in C. elegans, in flies, even in mammalian cells. Potassium ion channels that are mechanosensitive had been identified. But this couldn't be the touch receptor, for example, because activation and potassium going through cells would not activate the neuron, would do the opposite. So we were looking for a cation channel that is mechanically activated, and this we identified in the lab in 2010. I also want to go back to history just a little bit and ask, why did it take so long to identify these ion channels, just by comparing to what was done with other sensory systems? So the first transduction molecule that was identified was for vision, and rhodopsin, GPCR, was identified even before the era of molecular biology, in a way, by hardcore biochemistry. So just to get an idea, these receptors are present in this rod outer segment at a mind-boggling 50% of weight are the GPCRs themselves. So they were able to purify the protein first, sequence the protein, find out it's a G-protein coupled receptor. The very seminal work by Richard Axel, Linda Buck, and later on Charles Zucker and Reba identified similar G-protein coupled receptors by homology in taste and olfactory receptors, because the idea was that it was known that G-protein signaling was involved in these systems. So when we talk about mechanosensation or thermosensation for that matter, as I said, we knew there were ion channels, and unlike G-protein coupled receptors that at sequence level have strong homology to each other, ion channels really don't. If you line them all up, there's really not much in common with them other than that they have multiple transmembrane domain. So we knew that homology model was not going to work, and we had to have a functional assay to find them. So the outline of my talk today is quickly tell you how these piezo ion channels were identified, and then talk about the role of PIEZO2 in somatosensation and interoception. And then just the last few slides to take you away from neuroscience and tell you that PIEZO1 is playing an important role in various cell types beyond the nervous system. So the guy that identified piezo in my lab is Bertrand Coste, a postdoc in my lab who's in faculty in Marseille. He was with me in Oslo, but has returned back home. And he came in specifically to identify these ion channels. And he already was working on the RG neurons and had learned this very direct assay for mechanosensation. And the idea was he would do whole cell recording of cells while with a glass probe poking at the cell. And so you could see just this video what this looks like. You can see the patch on my right side, and with this glass probe, he simply comes and pokes the cell, and if this cell expresses a mechanically activated channel, then he would record this mechanically activated currents very easily. But after some failed attempts to try to identify this ion channel from sensory neurons, we took a step back and took a reductionist approach and said, why don't we work with easy to work with cell lines that we can just culture and incubator, and they easily grow. And the logic here was that although we were interested in touch and pain, all other mechanically activated cation channels in vertebrates were not identified, whether it was for hearing or sensing blood pressure, etc. So we said we'll find it in a boring cell line and then go back to in vivo and look at where it's expressed, and that'll tell us potential roles in vivo. So after screening a few cell lines, he found that Neuro2A cells, commonly used in neuroscience, actually had a robust mechanically activated currents. And then I won't go through details, but he made a list of candidates by expression, as well as the fact that they had 2 or more transmembrane domains, and made this list, about 300 candidates, and decided to knock them down one by one and see if he knocks them down by RNAi, if the currents stayed or not. So this is his epic slide that he spent a long time on. So what you're looking at is the maximal mechanically activated current from Neuro2A cells, and the top one in blue is the control, the tiny error bars. And then for each candidate that he thought might be the one, the first one being at top, he would knock it down, record, you wouldn't see any difference, move on to the next one. So these kind of error bars, and it's electrophysiology, so required that he spend about 2-3 days on each candidate. So after 11 months of getting negative data, he finally knocked down in red candidate number 72, and he saw a massive downregulation of these currents. So it was called FAM38A for unknown family 38 member A. He quickly renamed it PIEZO1 from piezi, Greek, meaning pressure. And then we got really excited when he did this experiment, which is getting the full-length PIEZO1 cDNA, and then overexpressing in naive HEC293 cells, and seeing, you can see the example on the left, and the massive upregulation or recording of mechanically activated currents. So it's not only necessary, but sufficient to induce mechanosensitivity. And PIEZO1 had a closely related sister, about 40% identical in protein, and this also had robust mechanically activated currents. If you look at it, the only difference is that the currents inactivate a little bit faster. Another very unique thing about this protein, which in retrospect, why overexpression screens wouldn't have worked very easily, is that it's a massive protein. So it's about 2,500 amino acids, PIEZO1 in mouse, and more remarkably, it has 38 transmembrane domains. So most ion channels have 2 to 6 transmembrane domains. This one has 38. It's more than any other protein containing transmembrane domains in your genome. On top of it, it comes together as a trimer for forming this monstrous, almost 1 million Dalton ion channel. Many labs, including Rod McKinnon's and Bailong Zhao and my lab together with Andrew Ward, have solved the structure of PIEZO1. This is the cryo-EM structure showing the 3 protomers, each in different color. All these ribbons are helices. So each protomer kind of extends into the membrane by themselves. They come together in the C-terminus to form the pore of the ion channel. Here is in the middle. And immediately what you notice is this awkward bend of the protein takes. And this, now Rod first suggested this, is that perhaps it's the mechanism of activation, that any flattening due to pressure on the membrane would cause activation of this ion channel. Indeed, his lab together with Schuring lab did this in liposomes, but very recently, Bailong Zhao, who trained with me and now is in Beijing, was actually able to get a structure of the flattened piezo, and it's quite remarkable. And here's the morphing of the 2 structures, presumably closed and open, although we don't know that. So as beautiful and remarkable as this looks, still there's a lot to do in this field, including trying to see what these ion channels look like in native plasma membranes, which we still do not know. Okay, so for the rest of the talk, I want to talk about the physiological relevance of these ion channels. And we have looked at this both from loss of function and gain of function mutations, and both in mice and humans. So we were ecstatic to see that PIEZO2, the sister molecule of what Bertrand cloned from Neuro2A cells, is highly expressed in these dorsal root ganglia somatosensory neurons that we're very fond of. And this is in situ hybridization, RNA-scope in situ hybridization, shown in brown of a section of a DRG neuron. You can see many neurons express it. I won't tell you details, mostly in low-threshold mechanoreceptors, but in some nociceptors as well. And in looking at the consequences of knocking out PIEZO2, we've used various Cree lines. I won't go into details, but I'll show you some of the phenotypes. And the first one I show is very rudimentary assay, but it's actually one of the first behaviors we've done with this mice, so I'm very fond of it. And again, not very sophisticated, but we call this tape on the back assay. And here's a wild-type mouse with a big white tape on its back. And as you can see, it doesn't really like it, tries to get it off. And you know, we think that the knockout is completely oblivious of this and is just walking around ignoring the tape. Again, we've done much more quantitative stuff, please believe me, but I just wanted to show you this. Kavli was not based on this assay. So I mentioned proprioception, and I mentioned a few days earlier that I believe proprioception is one of the most important senses that we have, but most people don't know about it. So this is how I can, you know, close my eyes and touch my nose. And I guess if you're very talented and practice, you can do things like this because of proprioception. And proprioception also appears to be strongly dependent on PIEZO2. So here's another mouse model with the kind of below lung lack of PIEZO2, and you can see the extreme uncoordination of these mice in the hind legs shown in this video that is characteristic of loss of proprioception. And for all our behavioral assays, we pair this with some in vivo or ex vivo recordings. And here you can see this is work done in our collaborator, Katie Wilkinson's lab. These are single proprioceptive endings being stretched and recorded. And you can see massive increase of firing when you stretch these endings. And in the knockout, 7 out of 8 saw nothing, a tiny bit in one of them. So both electrophysiologically and behaviorally, it seems like PIEZO2 is required for proprioception. So one of the more fascinating twist of this story is that my colleague, collaborator, and friend Alex Chesler and Karsten Bonneman at NIH actually identified human individuals that lack PIEZO2. And remarkably, they have the very similar phenotypes that we have described in the mouse. And so in a proprioceptive assay in humans that they developed, this individual who doesn't have any PIEZO2 functional in their genome is blindfolded, harnessed for her own protection, and asked to walk in a straight line. So I presume all of us would be able to do this without any difficulty, but as you can observe, she has very hard time doing this. So both touch and proprioception appear to be strongly deficient in individuals without PIEZO2 as well. How about pain? We know that acute pain is pretty normal both in mice and humans without PIEZO2, but we also were very interested in, as David talked about, the sensitization of pain, which is very important for chronic pain studies, and specifically tactile allodynia. Again, two complicated words, but very simple. It just means when touch becomes painful. So you experience this after sunburn or injury, that touch or just wearing a shirt becomes painful. But this is something that folks that suffer from neuropathic pain have it constantly, and there's really no great treatments for it. So Shweta Murthy in my lab and Alex's lab have shown that in mouse, all forms of tactile allodynia seem to be dependent on PIEZO2. And remarkably, Alex's lab has also shown that the humans without PIEZO2, capsaicin-induced allodynia is lacking in these individuals. This gives us hope that in a clinic, blocking piezo2 might be helpful for people who have this kind of various forms of neuropathic pain that are dependent on tactile allodynia. Okay, so just to kind of summarize this overall direction, so we've shown that piezo2 is required for touch and proprioception, tactile allodynia, and I didn't mention mechanical itch. So you know, a bug crawling on your back can cause this itch that is useful for you, and this seems to be dependent on PIEZO1, which I didn't show you before, but it is expressed in very few neurons in the RGs. So these are all what I call kind of exteroceptive communication with the outside world. But in addition, to DRGs innervating internal organs, there's a whole other system of the vagus nerve. Nodose neurons, for example, innervate many of the internal organs to gather both mechanical and chemical signals. And some of this you're conscious of, some of them you're not, but they play a very important role in homeostasis. And together with Steve Lieberliss's lab, and Alex Chesler's lab, we've been investigating the role of piezos in these internal organs, and I'm just going to summarize it here. For example, we've shown that you have these fibers innervating the lung, which monitor your breathing pattern and regulate breathing pattern. You're not conscious of this, but you actually have nerve fibers that innervate the aortic arch and monitor blood pressure. And again, if blood pressure increases, they work to reduce it through the autonomic nervous system. And these seem to depend both on PIEZO1 and 2 at the same time. And finally, if you drink too much coffee, you immediately feel that you have to go to the restroom. And this is because you can sense your bladder stretch, and you're very conscious of this. And we have shown, again, both in mice and humans, that without PIEZO2, this sensation is not present. And actually, the individuals without PIEZO2 learn to go to the restroom on a schedule because they cannot tell when they have to go, and when they do, it's too late. But I also wanted to make the point that this is a field that's still lots of unanswered questions. And just as one example, what is the role of mechanosensation in the gut, both in the intestine that David was talking about, but also for stomach stretch is still not known. Most of research on satiety, fullness, comes from nutrient sensing, which makes a lot of sense. But how much this stomach stretch that intuitively you feel when you have a big meal contributes to how much you eat, when you eat, and diabetes and obesity is not known, and we believe that studying these mechanosensors can tell us interesting new biology and physiology. So the last few minutes, I just want to tell you that, yes, we focused on sensory neurons, but many cells in your body experience, sense, and respond to mechanical force. So blood vessels experience shear stress, muscles are stretched, many cell types get indented, including sensory neurons and red blood cells. And the molecular identity of these sensors and their roles they play are still mostly not well studied. So this is just a summary of work done in my lab and many others on PIEZO1, the ion channel sister of PIEZO2 that is much more widely expressed. Shows, for example, that PIEZO1, both in humans and in mice, is required for blood vessel formation during embryogenesis. Others have shown that it actually is required for sensing bone density. And this is very interesting. For example, for decades it was known that astronauts who go into outer space and don't experience gravity, so no mechanical force on their bones, actually have reduced bone density. Together with Meta Patrick's lab at UC Irvine, we've shown that keratinocytes in the skin have PIEZO1, and they're required for cell migration after injury. But a lot of effort actually has come from this area that I didn't know anything about. Just the genetics took us a role of PIEZO1 in various, both red blood cells and immune cells, white blood cells. And I'll, again, show a few slides on this. A lot of this, again, started with human genetics. So there's a very rare condition called hereditary xerocytosis. And they knew they had dehydrated red blood cells. And we and others found that this is because they have a gain-of-function mutation in PIEZO1. More genetics, mainly in the mouse, suggest that loss of function of PIEZO1 in red blood cells causes overhydrated red blood cells. So now we think PIEZO1 plays an important role in hydration of red blood cells. I won't go much into detail, but inspired by the sickle cell literature that deformed red blood cells are protective against malaria, we've looked in the mouse and found that gain-of-function mutations of PIEZO1 offer some protection from malaria infection in mice. So after this, Shang Ma in the lab asked the question, similar to sickle cell, if this is the case, are there gain-of-function mutations in regions that are endemic for malaria? And he identified using genomics a very interesting allele, E756 delete, a deletion of one amino acid, that seems to have a pretty prevalent presence in people of African descent. So this just chart shows the prevalence of this allele. In the light blue is the wild type, and the gain-of-function is in purple color. And as you can see, this allele is pretty much not present in people of European descent, but 1 in 3 people of African descent are heterozygous for this allele, and this translates into pretty large numbers. 400 million people are carriers, and 40 million are homozygous at this allele. With these numbers, as you can imagine, this does not cause a disease, but given the phenotype-genotype association studies, these are kind of very useful tool to look at associations of how this subtle allele can associate with various conditions. So we're this is the formula that we're going back and forth on that's kind of become very useful for us. So I said the people who are actually diagnosed with hereditary xerocytosis, there's only a few hundred people worldwide, but they're deep phenotypes, so we can learn what goes right and wrong with them. And then we have mouse models. So this gain-of-function model that we've made is also Cre-dependent, so we can express it in a very specific cell type and find out what it does at a mechanistic level. But we also have this new powerful tool, which is then go to this subtle allele that's very prevalent and try to see if this might make a difference in the clinic. And so doing this, we've shown that gain-of-function PIEZO1 has potentially both advantages and disadvantages. I've talked about potential protection from malaria. More human work needs to be done before we say anything with confidence. But we've also found some potential disadvantages. For example, Shang has shown that this gain-of-function allele in another cell type, not red blood cells, causes overactive phagocytosis in macrophages, and this causes increased iron levels. Chronic levels of this is not good, of course, because it's toxic to many organs. And I'll just show one data of taking this and translating it into the larger clinical setting, where he's got about 300 individuals. He got blood cell samples from 300 individuals of African Americans. And as we expect, I told you 1 in 3 would have this allele, and this first graph just shows that. But when he starts looking at high iron levels in these individuals, you see the pie chart starts expanding towards the ones that have this gain-of-function allele. And if he looks at a clinical level of high iron levels, from these 300 individuals, every single one that had this high levels were all either heterozygous or homozygous for this allele. So they're 12x more likely to have these higher iron levels than non-carriers, so pretty significant. But much larger studies are required that I'm hoping clinicians will jump into and look at this more carefully. And again, we're excited to use this genotype-phenotype association to find out more instances where PIEZO1 is involved in different aspects of physiology and disease. Okay, I'm going to close the last 2 slides by saying that piezos are actually conserved across many species across evolution. They're not in bacteria or yeast, but they're found in various animals, including Drosophila, C. elegans, unicellular organisms, as well as plants. And just last year, Ali Reza Musavi, postdoc in my lab, looked at the role of piezo in plants. So plants also use mechanotransduction. For example, roots, when they encounter a harder surface, they have to either go through it or go around it. Ali Reza found that piezo in Arabidopsis is expressed right at the root tips where mechanosensing was thought to happen. And just one data slide showing that if he exposes Arabidopsis to harder gel-like material, the wild type, you don't see this very well, but they go right through the gel, so you don't see the roots, while the piezo mutants cannot seem to go through it, and they go around it, and it's focused on there, so you can see all the roots. So, but really, just an example of how amazing evolution is, that the channels that you have to sense touch seem to be conserved with those in plants to sense hardness of the surface that they have to penetrate. Okay, so PIEZO2 is the principal mechanosensor for many aspects of somatosensation and interoception. PIEZO1, on the other hand, seems to play an important role in non-neuronal cells. And again, I'm very excited of the studies that use both human and mouse genetics, providing a foothold into studying novel aspects of physiology and disease that depend on mechanotransduction that we didn't even think about. But for others who are interested, there's still mechanosensors to be found. As I said, this acute pain, hammer hitting your finger, how we sense it, we don't know. We've been trying to find it for the last 10 years and haven't yet, but we're still working on it. Finally, thanking all the current and alumni in the lab, as well as collaborators from all around the world that have helped over the last 20 years that I've run my lab, and of course funding, but NIH as well as the Howard Hughes Medical Institute and others. But thank you for your attention.
Thank you, Ardem, for this spectacular talk. We have time for questions. Okay, let's start with Soledad this time.
Thank you very much for the great presentation. So I have two quick questions. The first one is how distributed PIEZO is. So you showed that you found it, for example, in dorsal root ganglia. And my question is pointing to whether we get injured in some specific system, whether if it is distributed, then we would not lose mechanosensation, right? So that was the first question. And the second one is whether these cells that have PIEZO are innervating specific cells in the periphery or cells with specific properties.
We've actually had a miserable time exactly locating piezos. They're very expressed at low levels. This RNAscope in situ, which is very sensitive in situ, nicely shows the cell bodies that express it. In the periphery, where they exactly innervate, it's been tough. So we've even knocked in GFP at the end of PIEZO2, it's the fusion protein. And in some cases, we can follow it, and you can actually see beautiful, the proprioceptive endings of the nerve fiber wrapping around the muscle. But we haven't been able to look at in injury models, etc., to see how they change and all of that.
Hey, I have a question regarding the when touch becomes pain in inflammatory situations. So whether the inflammatory factors affect or alter these channels, that's one question. And the other question is like whether these 38 transmembrane, are they functionally relevant for this channel? All the 38. The
second part again?
Whether all the 38 transmembrane domains are functionally relevant for this channel.
Yeah. So the first question was the mechanism of tactile allodynia. We don't know the answer well, but we have hints that there's this central disinhibition model that touch neurons, mechanosensitive neurons, usually talk to the spinal cord centers of pain, but they're blocked. And when inflammation happens, this block is released so that then touch becomes painful. So you don't require a change of expression necessarily, just the circuit manipulation that causes this. And that seems to be the favorite hypothesis. With respect to the second part of this massive 38 transmembrane domains and what's required for sensing, again, we haven't made much headway there. Every time we've cut a few transmembrane domains off, the protein doesn't go to the plasma membrane. So, but although we did most of these experiments pre-structure, so maybe it's worth going back and doing more clever experiments to test these hypotheses.
Max, last question, please.
Yes, my question is about the recovery from inactivation of piezo, and whether this is related to the habituation to touch. You know what I mean? So how these channels recover from inactivation?
Again, a complicated answer. It seems like, first of all, overexpressing these channels are always what we call rapidly inactivating. I mean, they don't completely go back to zero, but most of the current is gone within 50 milliseconds. But in some cell lines which depend on piezo, their mechanosensitivity, the response stays the whole time you give the stimulus. And in vivo, this is also the same. So many of these fibers that I described that depend on piezo are actually slowly inactivating. So there must be some very interesting modulatory pathways that modify this inactivation kinetics, and it's used in the skin afferents for different uses, and we don't understand it yet. But this is actually one of the reasons why physiologists thought that touch would have at least 3 or 4, if not more, mechanosensors, because when they were recording from them in vivo, they had such different properties that no one thought one channel could account for most of these, and that seems to be the story we're going towards. And again, this modulation is going to be very important to figure out.
Thank you, Ardem, once again for this wonderful talk.
Thank you.